Welcome all of you. In this video, we will discuss about again nanosensors which able to detect pesticides and in a fruits. It is not a big deal. Like even the earlier, so many research has been done for detection pesticides and even any kind of contaminations like before consuming that fruits. But only thing is that those things are not portable. Like you cannot, like individually you can't do that, and even if you can do that, so you need to pay a high amount. So what recently people has done, as my in one of the earlier video, I'll talk about the SERS, which is surface enhanced Raman spectrum. So these SERS signals, as I told you, is very important, and sometime maybe I'll make a separate video on the SERS. So they utilize the SERS signals, and how they utilize the SERS signals. So actually, what they did, they make some silver nanoparticles, and then they use the silver nanoparticles and they immobilize on a glass surface or glass slide by flame. And once they immobilize it over there, then those silver nanoparticles are also coated with these small gold nanoparticles, like it's called the 2 nanometer or 4 nanometer or 5 nanometer of particles. So they coat over this, and then suppose they want to detect anything, so on a pesticide on any fruits, so they they detect they one of the pesticide which is called these ethyl pesticides, which different form of ethyl, which is even the some of the country, those pesticides are banned because those are poisonous for animals. But people are using it just for the insect free crop. They took the one of the apples and they bombarded that pesticide some particular locations, and then they use the cotton swab that area, put over in liquid suspensions, and take a drop of that and put over that glass surface, which is immobilized with the silver nanoparticles having the SERS called gold coated nanoparticles over the silver nanoparticles to increase the SERS signals. So they pour that drop from that sample, just a one drop, they pour that, and then they measure the signals in a spectrophotometers and in a spectrometer. Once they receive that, they find that pesticide signals, like as I told you in the SERS signal, what happening, the Raman signals. So each and every organic compound have their specific signals. So that pesticide also have their own lambda max. Lambda max is a wavelength which is absorbed at a maximum, that is called the lambda max. So that wavelength, once they detect that wavelength in a spectrometer, then people come to know, okay, that particular chemical moiety is present in that particular samples. So in that way detection is happening. So just imagine, like even for the warehouses where these kind of fruit has been stored, so people can detect what what level of the pesticide is. It is not only the qualitative, even it is a quantitative. And why it is a qualitative, you can understand, because yeah, it is yes or no. But why it is a quantitative? Because this is a lambda max, which is measured in the spectrophotometer. So intensity of that wavelength will determine the concentration of that pesticide. So suppose once they wipe with the cotton swab that particular pesticide area and they drop it there, and suppose in one case there is 100 molecule of pesticide, in another case suppose there is a 10,000 molecule of that. So per unit of area of that cotton swab, like having the higher number of pesticide where the concentration is high, so definitely the liquid suspension bombarded with that pesticide. So whenever that drop, we will take, suppose we will take 1 micrometer of drop. So that 1 micrometer drop, in case of 10,000 molecule, it is a higher concentration of pesticide, and other is less. So when they measure in a spectrophotometer, their lambda max also give a higher peak. So in that way they can measure this quantity also, quantity of that pesticide, and it is very handy. And apart from that, you can detect it within a time. So and yes, so some people can think of a spectrometer is very bigger machine. Yeah, those are the bigger machine also, but at the same time those spectrometers nowadays is a portable also. Yeah, like a bigger cell phone. like that, like about half an kg or 200 gram, not 200 gram, like about the 500 gram or 400 gram of weight of that particular device, handy device like mobile device. These are already available, so in that way people can detect concentration of pesticide over the fruits. So even in some of the peoples are in a market, they're claiming that we have very organic food range. So that thing can be detected easily whether it is a completely real organic, because if it is organic then it should not have some any kind of pesticide molecule. So it is a great step, but only thing is that the few reproducibility is one of the issues in this. So people are trying to optimize it to resolve this reproducibility, and apart from that, wide range of application, because those search signals can be perceived by they tested on the one particular pesticide, but how wide range of pesticide can be covered with that particular device is one of the questions. So for that, do we need to modify that silver nanoparticle, gold nanoparticle, or they can do some kind of generalized way, because these are the ligand and biomarker kind of interactions. So these are very specific to particular pesticide. So if you want to make any kind of generalizations where it can detect wide range of pesticide, so that things need to be development. But still it is a good benchmark study they have performed, and yeah, so in the future things maybe at home we can detect, like whatever the fruit we take like buy from the market, we can detect okay whether it's a that fruit is from the pesticide based or what. So we can do that in near future. So that's it for now, and if you need any brief explanations on that, so just DM me, and let's meet in the next video with some another topic. Till then, bye bye, take care.